After watching the last 10 minutes of the other membrane video, you have an overview of some of the characteristics of membrane proteins. I simply wanted to provide a bit more detail about this major component of biologic membranes. Specific to the membrane protein function, the previous video highlighted that membrane proteins can serve as transporters or channels, as well as receptors in cell membranes. However, there are a couple of additional functions of membrane proteins that occur quite frequently. Membrane proteins can function as anchors to either the extracellular matrix or to cytoplasmic components, or possibly to both. Additionally, membrane proteins can serve an enzymatic function, working to perform cellular processes that are necessary at the membrane. In addition to being categorized by their function, membrane proteins can also be categorized by the mechanism through which they associate with the membrane. There are integral membrane proteins, those proteins that associate with the hydrophobic portion of biologic membranes, and peripheral proteins, those proteins that most likely are associated with membrane proteins through protein-protein interactions. These protein-protein interactions are often non-covalent in their chemical makeup, and therefore they can be disrupted by the same things that disrupt other non-covalent interactions, specifically high salt, changes in pH, or changes in temperature. The integral membrane proteins come in multiple different flavors. They can be transmembrane proteins, as shown in the A part of this image. They can be monolayer associated alpha helices, as shown in the B portion of this image, or they can be linked directly, covalently, via lipids, and it's the lipids rather than the proteins that are going to associate with the hydrophobic portion of the membrane. In either case, all of these are integral membrane proteins, and in order to disrupt the association between integral membrane proteins and the other components, i.e. the lipids of biologic membranes, we need to use detergents. Like membrane lipids, detergents are amphipathic in nature, having a polar portion and a nonpolar portion. Because they have that same chemical character as membrane lipids, they are able to associate with the membrane lipids, as is shown in the lower right-hand side, and they are also able to associate with the hydrophobic portion of membrane proteins, as shown in the lower left portion of this second image. Disrupting biologic membranes with detergents will therefore liberate both or separate the membrane lipids from the membrane proteins, allowing you to isolate membrane proteins from membrane lipids. Regardless of how the proteins associate with biologic membranes, there are a number of proteins that do associate with the membrane on the cytoplasmic side of the membrane. That combination of proteins that sort of line up or underlie the cell membrane on the cytoplasmic face are referred to as the cell cortex. The cell cortex is comprised of a number of different proteins that associate both integrally and peripherally with the membrane to provide both structural support and sites for proteins to anchor to the cytoplasmic components. Just like membrane lipid mobility can be modulated by the environment in which membrane lipids find themselves, it's also possible to limit mobility of membrane proteins. One mechanism through which this can occur is by embedding membrane proteins in lipid rafts. Recall from our previous discussion that lipid rafts are less fluid regions of the membrane, and they're less fluid because of a high concentration of cholesterol, because the lipids that are in this region also have unsaturated fatty acid tails and longer fatty acid tails. In addition to embedding proteins in lipid rafts, it's also possible to limit protein mobility by anchoring membrane proteins to either the cell cortex on the cytoplasmic face of the membrane or to the extracellular matrix on the extracellular face of the membrane. If these membrane proteins are bound to less mobile proteins themselves, then the membrane proteins will be less likely to move around within the cell membrane. It's also possible to have membrane proteins anchor to other membrane proteins, as shown in panel C. If these proteins are associating with one another, you can imagine that these proteins for both cells, or on the surface of both cells, would be less able to move around because of this cell-to-cell contact. Finally, cell-to-cell contact is another mechanism through which protein mobility can be limited. There are specific types of cell-to-cell junctions referred to as tight junctions that preclude the ability of molecules to move between adjacent cells because of the riveting, if you will, of two adjacent cells to one another. That tight riveting of two adjacent cells to one another sets up a situation where there are two distinct environments within this cell membrane. There is an apical surface that's exposed to one type of environment and a basolateral surface that's associated with another. The tight cell-cell junctions prevent the movement of membrane proteins on the apical surface from moving to the basolateral, and similarly, those proteins on the basolateral surface are precluded from moving to the apical surface. So in this case, a cell can have two very distinct membrane domains where mobility, protein mobility, and indeed lipid mobility is limited to within a domain and not between those two domains. Finally, I'd like to reiterate a bit of information about membrane carbohydrates, both the location and the function of this very important membrane component. Recall that membrane carbohydrates are localized to the extracellular leaflet of the plasma membrane to create what I like to refer to as a sugar coat on the surface of all cells. Of course, the thickness of this sugar coat is going to vary between cell type depending upon a specific cell's function. This sugar coat is referred to as the glycocalyx and is comprised of cell surface glycolipids and both transmembrane and peripheral glycoproteins that have been decorated with carbohydrates during their synthesis. This glycocalyx performs a number of very important functions, as the previous video described. These include adhesion, or sticking to other cells, and communication, both of which play a role in immune system function. I want to provide a bit more detail about how the glycocalyx works to support immune system function. It's important in allowing white blood cells to both slow down within the bloodstream and to leave the bloodstream at a site of injury or infection. In this diagram, a neutrophil is careening very quickly through the bloodstream, although there is an infection in the interstitial space nearby. How is this neutrophil going to know that there is an infection nearby? Much less, how is the neutrophil going to leave since it's moving so quickly past this site of infection? This is dependent upon an interaction, a specific interaction between a carbohydrate on the surface of the neutrophil and a protein receptor that is being expressed on the surface of the specialized epithelial cells that line the bloodstream. When an infection is nearby, those specialized epithelial cells begin to express or display a protein receptor for a neutrophil carbohydrate. A few terms. The specialized epithelial cells that line blood vessels are known as endothelial cells, and the proteins that interact with carbohydrates, the protein receptors, are referred to as lectins. The association between the endothelial cell lectin and the neutrophil carbohydrate slows the neutrophil down and signals that there's trouble nearby. It's almost as though the carbohydrate coat on the neutrophil is sticky with respect to the lectin receptors, and this slowing down allows the neutrophil to begin to remodel its cytoskeleton inside of its cell so that it can deform and squeeze between two adjacent endothelial cells. The process of leaving the bloodstream is known as extravasation. Extravasation, the process of leaving the bloodstream, necessitates moving through the endothelial cell layer. This crawling through the layer of cells is called diapedesis. Here are two additional terms for you. Another function of the glycocalyx that the previous video neglected to mention is that of protection. This is very important for chondrocytes, for example, in joints that must resist compressive and jarring forces when a person moves. Specific to blood cells like the neutrophil that's shown in the diagram, as I said, blood cells are careening very quickly through the bloodstream, propelled by cardiac contraction. As the heart beats, the blood and the cells in it are propelled very forcefully, jarring against the blood vessel walls. The glycocalyx associated with blood cells serves to protect against the shear forces they encounter in the bloodstream.